So Amber, you think that you can be a scientist?
I do think I can be a scientist.
Let me tell you, this job is not cut out for everyone. So before I even tell you about this once-in-a-lifetime opportunity, I'm going to need you to prove to me that you can actually express recombinant protein in bacteria.
Look at this stuff. Isn't it neat? Wouldn't you think their collection's complete? Wouldn't you think that this lab has everything? Look at this water bath. It kind of smells bad. How many incubators can one lab even have? Looking around here, you think, sure, they've got everything. They've got gadgets and gizmos aplenty. They've got micro pipettes galore. You want inoculating loops? They've got 20. But who cares? No big deal. I want more. I want to be where the scientists are. I want to see them experiment, walking around in those, what's that word again? Oh, lab coats. Clicking a pen, you don't get too far. Scientists require a detailed protocol, writing things down in those, what's that word again? Oh, lab notes. Up where they walk and never run, up where they stay all day in the lab, experimenting. Wish I could be part of that lab.
Hey, are you even paying attention?
Yeah. Recombinant proteins and bacteria and stuff.
Well, get a move on it then.
Oh, God.
So recombinant protein is a foreign DNA, and we are going to place it into another organism such as E. coli. We are looking at the protein retinoblastoma N-terminus because it is suspected that it will inhibit helicase activity, which will stop cancer from proliferating. Retinoblastoma protein N-terminus is being looked at as an inhibitor for helicases. In cancer cells, helicases being inhibited will stop the proliferation so that the cancer cannot increase rapidly throughout your body. So this is a plasmid. This plasmid is going to be used to express our protein, RBN, which is tagged to GST, which will later be purified in a later step. We have a reporter gene ampicillin resistance, and this is going to help us see if our transformation was successful or not. So we're going to take a BL21 competent E. coli. This bacteria is easily transformed. So we're going to heat shock our mixture of our plasmid and our E. coli in order to obtain pores in our bacteria. Our plasmid is then going to enter our bacteria. Then we are going to close up those pores that we just created by icing it.
Samantha? Do you want to transform bacteria? We'll put our plasmid into them. First we'll stick them in a water bath, but not for long or else they'll all be dead. Then we'll plunge them in some ice. It'll be so nice. It'll put pores into them. Do you want to transform bacteria? It's BL21 competent.
Go away, Amber.
E. coli. Transform bacteria. So what we want to start with is growing up whoever we are going to transform into to log phase. And what log phase is the best phase to transform things in. Is because they're rapidly dividing. It's like the sex-crazed teenager stage of bacteria because they're just like, hey, let's get together and divide. So, to do a transformation, we're going to take about 1 ml of our bacterial sample, so 1,000 microliters of it. Just like this. Place it into our tube that we're going to mix our plasmid in. So we're going to take about 2 microliters of our plasmid, which should be left on ice. And just put that into our E. coli. Kind of mix them up. So we're going to incubate that on ice to get them nice and cold for about 20 minutes. So let's just say 20 minutes has magically gone by. We're going to bring our ice over to our water bath so we can perform our heat shock. So our water bath is set at 42 degrees. You just open this up, heat shock this for about 90 seconds. 90 seconds went by, and immediately stick it back in ice. That's how you're gonna get your pores to open and your pores to close with your plasmid inside your bacteria. So once you've done your transformation and your heat shock, we're going to plate our bacteria with our plasmid solution onto an LB plate and onto an LB with ampicillin plates. So once we have our E. coli with our plasmid solution, we are going to plate it on an LB and an LB amp plate. So you'll take about 100 microliters and you'll plate it on each. So to keep everything aseptic, you want to take your spreader that has been dipped in alcohol and light it on fire, because who doesn't like fire? So you're going to let it cool a little bit so that way you don't kill all your bacteria. So cool, cool, cool. Alright, so once it's cool, you're just going to spread like so all over the plates. Done. And you'll do the same for this one, like so. Alright, so these are going to end up going into the incubator set at 37 degrees for overnight. That way they can grow, they can get happy, and you should get something like this. So on an LB plate, you're going to have all the bacteria growth. This is bacteria that contains your plasmid, that doesn't contain your plasmid, so you're just going to see a lawn, and you're not going to be able to get countable colonies. On this plate, however, our LB ampicillin plate, the only bacteria that grows is the bacteria that contains your plasmid. So these are the bacteria that we're going to want to take from in order to express our recombinant protein. So what we do when we're ready to express our recombinant protein is we're going to take a colony from this plate, swish it around in some LB broth, and grow it up into an overnight culture. And the best way to do this is to have LB broth that also contains ampicillin, so you don't get any contamination from any other bacteria, and you're just getting your bacteria with your plasmid. So what you'll do is you'll grow that up overnight. And then if it's still not at the right concentration of cells, which you would check by taking an optical density, you'll just continue to grow it until it's at the concentration that you need for any other experiments. So,
In order to fully express our recombinant protein, RBN, we must use an inducer molecule such as IPTG. IPTG will bind to the promoter region, allowing for transcription and translation to occur. This acts as a bacterial inducer, which will then allow us to fully express our protein of interest.
So you actually transform bacteria. Good job, I guess.
yeah, I think it went really well. So what about this once in a lifetime experiment thing?
Right, about that. You already did that. I just didn't want to transform the bacteria myself, so thanks.
Really?